Again, this is kind of a somewhat tricky topic for a lot of people, so I decided to split it up into a couple videos to hopefully make it a little bit more manageable and break it down. So this first video is going to cover amino acids, just a little bit of background information, their basic structure, and I'm also going to cover for you how I memorize the amino acids. They can be tricky, but it's just something, if you're in the science field, something you got to do. But I think I have a couple tips or tricks that worked for me that might help some of you guys too. So this is my contact information. If you have any questions or anything like that, feel free to shoot me an email, and I'll either work with you one-on-one or just hopefully I can answer your questions. Okay, so amino acids. By now you probably know what they are. They're the building blocks of proteins or monomers, and they all have the same general structure, which, as you can see here, consists of amino group, a central central carbon, and then a carboxyl group over here, or carboxylic acid. What makes them very unique is they all have a side chain, which is variable, designated as the R group. But we can go ahead and use these. This is what we're going to use to find our isoelectric point. They're also classified by their R chain, which I think we'll see here. So this is all 20 amino acids. There's technically a 21st amino acid, but you're not going to really need to know that. But there's a lot of ways to separate them. You can separate them into essential versus non-essential. Non-essential amino acids are non-essential because we can produce them in our own bodies. Essential ones we need to get from our diet. But there's non-polar, polar amino acids, and then the charged ones over here. So these ones have a negative charge, aspartic acid or aspartate, and glutamic acid or glutamate. Positive is going to be lysine, arginine, histidine. And then all these are non-polar because they don't have any polar groups in their respective side chains. Sorry. The one thing to note is cysteine. Sometimes, depending on the textbook, that can be classified as polar uncharged. So just kind of be cognizant of that and kind of defer to whatever your professor tells you. Okay, so here's how I remembered them. First of all, the couple special things to note are some of the exceptions. One of them is that glycine here is achiral. It's the only amino acid that's achiral, meaning it doesn't have four unique groups attached to the central carbon. You should know that from organic chemistry, but that makes it kind of an exception or unique, so that's something to note. Another exception is going to be cysteine. Like I said, it's sometimes classified as polar, so make sure you defer to whatever your professor tells you. But it also is important in forming disulfide bridges, which is critical in the tertiary structure and the way proteins fold on themselves. Another one to note is proline, which kind of is an exception to the structure because it forms a ring with that amino group. Yes, I think that's all I wanted to cover with the exceptions. Okay, so unfortunately for the non-polar amino acids, I don't really have a trick for memorizing them. I will say that it's easy for me to group them in my head. Glycine, I remember because it's kind of, I always think of glycine as like the first amino acid, just because it's so simple. It's just basically this backbone structure with the carboxylic acid and the amine group, and then just two hydrogens. So it's pretty much just the most straightforward, simple amino acid. Alanine, same thing, just has a methyl group here. Valine, you start to get a little complex, but valine, think of V, it starts to form a V. And then if you jump down here to leucine, leucine looks just like valine, except there's one extra, the carbon chain is a little bit longer, but it still has that V. And then this is neat, conveniently, isoleucine right next to it looks just like leucine, except it's an isomer, and this methyl group has jumped up here. So these two, think of them together, just same structure, except one of the methyls has moved. Cysteine, I also remember because this sulfur group helps it form disulfide bridges. And then methionine, I remember that because they both have a sulfur group, so I pair those together. So basically what I'm getting at is I remember the exceptions. Cysteine, which always leads me to remember methionine. Glycine, because achiral, which leads me to remember alanine, which then in turn I can remember valine. These two I pair together, leucine and isoleucine. And then these are pretty straightforward, frankly. Remembering the aromatic ring side chains isn't that bad because, but that goes without saying, they're very unique and large side chains. Okay, so that's my sort of trick for nonpolar. Whatever works for you guys, but that's kind of what I use to remember the nonpolar ones. The polar ones, I did come up with this little I guess, acronym STAG. So it's STTAG, so obviously a little bit of an awkward or not completely accurate spelling, but I mean, it just goes right down the line: serine, threonine, tyrosine, asparagine, and glutamine. One thing to note about these, asparagine and glutamine, is that they're very similar to aspartic acid and glutamic acid, or aspartate and glutamate, whatever you want to call them. They're pretty much identical in structure, except these have an amine group, while their negatively charged counterparts have a carboxylic acid in the side chain. Tyrosine, kind of easy to remember. I don't know why, but like I just think the O, tyrosine, makes me think of the aromatic side chain as well. Again, kind of weird. Threonine, I remember it's because there's a CH3 or a methyl group in the side chain, along with an alcohol with the O. So that's just kind of how my head categorizes them. Moving on to the positively charged amino acids, LAH, or LA, is my kind of acronym for remembering them. All of these are going to have, well, it goes without saying, they have a positive charge due to this, like, I guess, I don't know, you can call it ammonium, I guess, or it's an amine group it's a protonated amine group, but all of those are going to have a positive charge on them. So those kind of are easy to remember. They all have really long side chains, and then histidine is kind of interesting because it again has a ring. Then that brings us to the negatively charged side chains, and I do have an acronym for that one as well. I go ED, but this one obviously is not based off of, I guess you could do AG as well, but this is based off of their one-letter abbreviations, which again, going back, all of amino acids have a one-letter abbreviation, which I highly, highly suggest you know, because when you're given a polypeptide, sometimes it'll just say like A, M, F, R, H, D, and you have to like know what those represent. So like I said before, these are very similar to asparagine and glutamine. I remember the difference because asparagine and glutamine, it has well, this one actually has amine in it, and this one is just ene, like reminds me of amine, where this one actually has a carboxylic acid in place. Okay, but that wraps up how I memorized the 20 amino acids. Hopefully some of these study tips kind of helped you a little bit, but moving into the next video, we'll work through how to find the isoelectric point. Thanks for watching, and make sure you stay in touch with ASC.